And our next presentation is going to be by Robert Coenraads. And Robert's a good friend of mine. He just lives a couple of suburbs away and attends our study group in Sydney. And Robert's got a doctorate in geology and gemology, and he's a wonderful person to go for a bushwalk with. You know, he can paint the landscape as it looked 230 million years ago, and you really feel like you're actually there. So I really appreciate Robert. And so he's going to do the second part of his presentation. The first time we had this 24-hour Zoom meeting, he did the first part. And Robert's presentation is on a group of papers dealing with the origin of life on Earth. And he just puts this little footnote in here for us. Although the revelators have made it perfectly clear that within a few short years, many of our statements regarding the physical sciences will stand in need of revision. These papers provide a fascinating and vital scaffolding for the spiritual message in the remainder of the book. These papers deny fundamentalist dogma, heal the rift between religionists and scientists, and allow all to partake equally in the book's spiritual message. Much detail remains unrevealed so that today's and future scientists may enjoy the thrill of uncovering the true elegance and the sophistication of the life carriers planned for Urantia. So with that, I'll hand you over to Robert Coenraads.
Hello, everybody. It's wonderful to have been here all this morning, and I'll now share my screen with you all. We're big, and away we go. We're going very quickly now because we saw this before. We saw the birth of the Earth. Please don't read, just enjoy these lovely images as I go. We saw the Earth forming in the Hadean period. And then after that, the wonderful book, the vast stone book of world record, which the geologists work from the bottom up, starting off with the beginnings of life in the Precambrian, and then the most marvelous Cambrian comes in, life in the sea. Silurian, the first plant life on a miserable bare earth. In the Devonian, the fish develop. Life moves on to the land. The first lungfish come out. And then we get the wonderful Carboniferous, the time of the earth covered with forests, giant insects and oxygen levels up to 30%. The Permian. Then comes the dinosaurs, the Triassic, the Jurassic, the Cretaceous. And then into the Cenozoic, we get the flowering plants, and the earth became colourful. And in that time, then we had the appearance of the mammals. And then we have the mammals, and of course, the appearance of atomically modern humans. Now, let's just have a look at evolution. The four basic viewpoints that I sort of detect around today: the scientific method, which I know and understand well, pure observation of the world around us in an attempt to understand processes without trying to ascribe a cause. Now, that's what I do. But now, number two, the liberal religious viewpoint, that includes Catholic and other major denominations. They believe this, and Pope Pius the 12th stated God's goals are achieved through natural evolution as described by modern science. But the individual soul is a direct creation by God and not the product of purely material forces. And a great famous Jesuit paleontologist, Pierre Teilhard de Chardin, defines God-directed evolution, or orthogenesis, in which man will eventually evolve to the omega point of union with the Creator. Now, I think that's very astute, because of course in number 3, the Urantia teachings, we have all of the above and with far more detail provided. Teachings about God as compatible with the modern scientific understanding, and of course free will means we are our own masters. And then number 4 is the fundamentalist religious dogma, which has to be done battle with, and that is that the world was created by God in 7 days. Now, if we have a look at Genesis chapter 1, you'll see that I've just put it up here, and we won't go through it, but to me, as a scientist, I just see the creation story in there. I see evolution. There's light, there's day and night. Look at the yellow words here. Dry land appears. I can move my cursor even, maybe. I don't know whether you can see that. Then the grass, then the trees. There seems to be, theologists think there's 2 stories in the creation story there, because down at chapter 1:16, God makes 2 great lights. The lesser to rule, the greater light to rule the day, and the lesser light to rule the night. He made the stars also. So we get a bit of a repeat, and then God said, "Let the waters bring forth abundantly moving creatures that hath life," and then the great whales, and then the winged fowl, and then the cattle and the beast. And then let us make man in our own image, in our own likeness. So God created man in his own image. I find it quite wonderful. Talking about likeness, these are the few words that create great battles. After our likeness, and the other ones are, here we go, be fruitful, multiply, and replenish the earth, and subdue it, and have domination over the fish of the sea, and over the fowl of the air, and over every living thing that moveth on the earth. So here in just 2 sentences, we can see a lot of problems being brought out by fundamentalists for humankind. But anyway, we don't quite want to go there on this occasion. I want to now have a look in detail at some of the mechanisms of the life carriers, because of course, as Trevor read out, those mechanisms are unrevealed because we have to discover them. Now, we're going to go quickly through some of these wonderful mechanisms. We won't spend time on this slide listing the mechanisms, because I have them on the following slide. But here, you can see our Earth 150 million years ago, and you can see the plates, and here they are separating into the current configuration. And of course, the animals on the plates went off on their own separate rafts. You can see where it happens. The big cats, the lion, the tiger, and the panther, they went off on their separate rafts. The panther of South America, the tiger of India, the lion of Africa. And 150 million years passed. And if you can see on the time scale about to come up there, the 150 million years is a very, very tiny amount of time, as shown by that yellow arrow at the top for the overall time scale. But in that short time period, the big cats did the following. They evolved into 3 separate species of Panthera: Leo, Onca, and Tigris. You can see them there, quite different, and different species means that they can't reproduce anymore. after 150 million years, but they certainly came from the same common ancestor. Now, why did they evolve? Well, here, try and have a look at this picture now of the tiger with its stripes, and you'll notice the camouflage benefit of this tiger moving through the long grass there. So that's evolution evolves due to the influence of the surrounding environment. Now let's have a look at the forelimb here. This is quite brilliant, really. Now, in the design, the master plan, you can see all the bones of the forelimb are identical. It's only that some are shrunken, some have grown large. See the fingers of the bat, for example, compared to the human on the left. Go to the next slide here. You can see the human, and the bones are coloured in, the ones that match. And you can see the digits of the bat and the digits of the human, and you can see the digits of the whale that have become wrapped in a rubbery flipper. The bat has become, the digits have become wrapped in a wonderful elastic membrane that enables it to fly. And of course, we've got our wonderfully dexterous hand and opposing thumb, which is far better than the cat next to us there. So that is evolution taking place before our eyes. Here we have the Hawaiian honey creepers, which Jerry will be intimately familiar, and they are the same species that has evolved beaks for different purposes. You can see they've short, fat, thick beaks for grasping, and long, thin beaks for entering deep into flowers and sucking out honey. So many things as the environment pushes. Look here at the woolly mammoth. During the Ice Age, the environment, the cold, the ice, forced the common ancestor of the elephant and the woolly mammoth, the one that happened to be living in the colder area, to evolve fur and to keep warm. Now, likewise, the Neanderthal and the human, Homo sapiens, atomically modern human, you can see that the Neanderthal is a cold Ice Age adapted human being, and the text there indicates the differences, which includes a stockier build and a larger head, stronger jaw, all with the aim of keeping the heat in and to undergo the trying times of having to live in an ice age, if you can imagine how that would be. And we have a few genes in us, the red hair and the prominent nose and various other things there, which we see. Now, that convergent evolution is a very interesting thing too. If creatures are going to fly in the air, many different species, families, and orders can go into the air, and they all look a little bit similar in the fact that they have wings and the ability to use air currents. You can see the falcon, the bat, and the pterodactyl. Similarly, when creatures return to the ocean, when environment sort of conditions encourage them to go back to the ocean, they developed around their digits the leathery flipper, and they got a nice body line and a tail, and they were able to adapt to the ocean. Of course, the dolphin and the Kronosaurus have to come to the surface to breathe, because they're not fish. They are land animals that have returned to the sea. Now, mutualism is very interesting, because that's interspecific cooperation between two organisms, in this case, the the hummingbird and the flower, two entirely different kingdoms, in fact, yet they have adapted to one another. The flower needs the bird as much as the bird needs the flower for food. There is the bee and the flower, both needing one another as well for pollination and food. Here's some interesting mutualism with the Egyptian plover cleaning the Nile River crocodile's mouth. Now, I don't think any other animal, apart from that black and white marked plover, can enter the mouth of a crocodile like that. The oxpecker birds on the top of this impala sort of pick off the ticks and other animals. And here we have an adaptation in the polar region. All these animals that migrated to the polar region, they adapted very similar features for camouflage, thick fur and fatty layer under the skin and other things like that, which let them survive in that environment. Although sometimes the camouflage doesn't always work in this particular case. Now, here we have the orchid mantis evolved to live in this orchid, and I bet you can't even see the mantis in the orchid there, just lying in wait for some unsuspecting bee or something else. The head is down in the bottom right there, and you can see the two front appendages just below that. On the other hand, the blue poison dart frog, the one that the South Americans rub their darts on to get that toxic effect, it's doing the exact opposite. It wants all its potential assailants to see brilliantly and exactly who and what it is, and dare them to even think about eating it. Mimicry. Here we have two snakes of entirely different families. That's not even species, but families. And look, they've evolved. The non-venomous coral snake has evolved the similar markings to the venomous Mayan coral snakes to keep it safe at all times, because no respectable falcon would dare come down and attempt either one of those, knowing that one might be venomous or one mightn't. So these are such wonderful examples in nature that the life carriers have put in motion. Now, here's the snakehead caterpillar. This will take a little bit of getting used to. That the snakehead is the tail of the caterpillar. The head of the caterpillar is on the branch above. And you can see another one here more obviously showing the head of the caterpillar on the left with its legs, and then it can raise its tail and it can pump fluids into it to give this snake-like head. And I mean, that's absolutely brilliant, even to the gleam on the eyeballs as a defensive mechanism. Now, no other creature is going to take a bite at that. It would if it thought it was a caterpillar, but thinking it might be a snake, it's certainly not going to come near that inoffensive caterpillar. So this is a very successful thing, and various caterpillars try it. That's the back end of another caterpillar, although some more realistic than others. In this case, it looks a bit like a pump-up pool toy. But nevertheless, obviously in the natural environment, these tails of caterpillars are well enough camouflaged to look like a snake that they are highly effective. The electric eel is practically blind. You can see its tiny little eye. lives in the Amazon, which hasn't got very good visibility, but it can discharge 900 volts. If anything comes near it or steps on it, it can kill a horse or a human. So these adaptations are really brilliant, and I will just delve into the octopus here, because I find its camouflage most amazing. You can see it moving into a spot on the left and then settling down and camouflaging. And it's interesting how it works. It's got these melanophore cells that can actually contract pigments into the middle of them. So if you have a look in this diagram, you can see a cross section through the skin of an octopus, and you can see the black spots in it would be dark melanophores, and these can be contracted in this star-like pattern. The muscles condense down and shrink these things, and then the next layer down, the red-orange layer there, can show through. And likewise, the red-orange layer can be contracted to make yellow, and this all happens automatically, of course, as you can see with this this wonderful chameleon exercise as the chameleon crosses the sunglasses here, and automatically, without even thinking about it, it just adapts to the appropriate background, so you may not notice it at all. And it can do this. It can really activate itself like that, or it can practically be black and white as need be. Truly amazing. And the same technique below the skin, you can see the three primary colors that can be contracted or expanded, so they can be combined in whatever arrangement. Truly wonderful indeed. Now, humans have likewise adapted. Here we've got the global map of indigenous skin colors. So that's indigenous. That's before we all started moving around the place. And you can see near the equator there, where humans evolved 300,000 years ago, they have very dark skins, and you can see the skin color lightening on either way towards the poles, where the skin is needed to be fully pale and transparent to accept that that's important sunlight. So I find that very interesting. Humans came to the Americas 10 to 15,000 years ago, and they haven't quite gotten as dark as needed in the equatorial regions as yet. And 70,000 years ago in Australia, the Aborigines are fully adapted to the strong sunlight that we experience here and get skin cancer from. This is a wonderful artwork, Human A by Angela Das, and here she's taken the respective skin color, I think off the side of the nose or cheek, and used it as a background for the photographs here. And she's done thousands of these. It's just a wonderful thing, showing the diversity in color, skin color range in the human being. So the life carriers, let's listen to the life carriers. The majority of inhabited worlds are peopled in accordance with established techniques. On such spheres, the life carriers are afforded little leeway in their plans for life implantation. But about 1 world in 10 is designated as a decimal planet and assigned to the special registry of the life carriers. And on such planets, we are permitted to undertake certain life experiments in an effort to modify or possibly improve the standard universe types of living beings. Most fascinating indeed. Most fascinating indeed. And so let's see what they might have organized for our planet here. Now, recently, we've discovered horizontal gene transfer. And if you have a look at these cells here, this is a series going from top to bottom. You can see number 1 is the donor cell, and it's got stuff inside it. It's got DNA, which we'll come to a little bit later. And then with its pilus number labeled 4 there, it comes together with another bacteria and shoots the red material, which has multiplied, which has made a copy of itself, and that copy passes through the pilus into the second cell. So you might say, well, why is the first cell doing that? It's passed on something to the second cell, and then you've got 2 identical copies which can then reproduce. So advantages conferred by years of evolution can be immediately transferred to another individual. And there we see it happening on the bacterial level. And how clever is that? You know, maybe this is something the life carriers have thought of to get things happening quickly. Very exciting indeed. But there are problems in God's plan. There's the Zika carrying mosquito. Now, I'm sure that this presents problems for the fundamentalists, just coming back to them briefly, because everything is created in its kind. Those 3 words in Genesis chapter 1, in its kind, means that God has created the butterfly and painted the patterns. God has created the caterpillar and the falcon, but therefore God has also created the mosquito, and therefore God has also created the E. coli bacteria, the meningococcus bacteria, the golden staph, and heaven forbid the Ebola, which we really want to avoid at all costs. So unfortunately, in this plan, a donor bacteria can develop antibiotic resistance. Here we see the fabulous thing happening again. And number 2 would be the bacteria with no resistance to a lovely antibiotic that we as humans have made. And then the first bacteria comes along, passes along the resistance using its pilus. And then we have years and years of evolved antibiotic resistance transferred to other individuals. And we have trouble. We have to work very hard. Not that it's past us to be able to do that. But here, talking more about the life carriers, after the life patterns have been formulated and the material organizations have been duly completed, the supermaterial forces concerned in life propagation become forthwith active, and life is existent. Whereupon the life carriers are immediately returned to their normal mid-phase of personality existence, in which state they can manipulate the living units and maneuver the evolving organisms, even though they are shorn of all ability to organize new patterns of living matter. So in other words, we do get some kind of implantation. We do get the life starting off in those areas and evolving much as you saw in the early slides. The life carriers do that. And after organic evolution has run a certain course and free will of the human type has appeared in the highest evolving organisms, so that's whatever organism gets the free will first, the life carriers must either leave the planet or take renunciation vows. That is, they must pledge themselves to refrain from all attempts to further to influence the course of organic evolution. Now, can you imagine how frustrating that must be for a life carrier to sit there and watch E. coli, Ebola, well, current COVID-19, and sit there, twiddle their fingers and think, oh, you know, that that plan we had for that horizontal gene transfer, look what it's doing now. damn it, and we're not allowed to fiddle. No, we've got our free will. And how important is our free will? That really illustrates it right then and there, that we do have the free will. Nowadays, we have the free will. Do we spend our earnings, our national product on armaments and defence and political advertising, etc., or do we spend it on medicine and education and hospitals and public services? There you go. It's all in our capability. Anyway, of course, DNA and all of that kind of stuff was very little known at the time of the writing of the book. So all of these wonderful life carrier things could not be told to us. But with the little bit of time left, let's just have a little. I'm going to have a little bit of fun and go into these genes. It's only a decade or two old, some of this study here, and show you how much fun it is and what's happened, and how the life carriers work in this little bit of detail. So here we have the human cell, and coiled up in the nucleus, we can see our genetic material there, a bit like an old telephone cable, and wrapped up in those are the chromosomes. We have 23 pairs, so that's 46 of them, and 22 of them are autosomes in our nucleus, and then the 23rd pair are the sex chromosomes. You can see them after the ones numbered 1 to 22, numbered according to their gene length, the chromosome length. You've got the sex chromosomes, a long Y, a long X and a short Y. And so uncoiling those in the DNA, you can see that there are 4 nucleotides there: adenine, guanine, cytosine, and thymine. You can see them there, A, G, C, T. And just to illustrate how simple these basic building blocks are, that's their chemical formula there. They're nothing more than chains and loops of oxygen, hydrogen, nitrogen, and carbon, very similar looking, and then they stack up in these genes. So that's inanimate life doing such amazingly wonderful and adaptive things that we've barely scraped the surface of our understanding. So now let's have a little bit of fun, because maybe the encouragement, it's a lot of fun to have your genes tested and look into your past, and it can even tie in with our Urantia part of the Urantia book too. But let's get a bit of background in here, if you'll bear with me. And I've got the family tree here with U down the bottom. And I'm now talking about the autosomal DNA. That's the majority of the DNA in the nucleus. And you can see that your parents there, going up one from you, contribute 50% each of their genes to your nuclei. The grandparents are 25%. The great-grandparents are 12.5%. And the great-great-grandparents, some of you may have even been lucky to know those, or there's getting more and more of them. That's only 6%, then 3%, and then 1.5% in the top row. So that top row at 1.5%, that's the limit of the autosomal DNA test. So if you go and pay your $50, and it's becoming cheaper all the time, you get information going back on all of those ancestors, looking back there in that chart. So here we go. There's you at the bottom again. And I just want to show you how quickly these great-grandparents increase in number. So counting back, that's 1, 2, 3, 4, 5, 6, 7, 8 right at the top row. Look at that for 128 great-grandparents. Now go back 15 generations and you have 32,768 of them in 15 generations, sorry. Now, in about the year 1263, we reach 1 billion ancestors. And so this is how we know for sure that we all descend from the same ancestor multiple times. That is, the number of our ancestors quickly exceeds the world population. So I'll show you, so you can imagine those numbers going back there. So here on this graph, you're coming back from 2000, and you can see the size of your tree increasing in numbers. And that's a logarithmic scale, by the way, on the left. So you can see your numbers going up. And then on the same logarithmic scale, you can see the world population, which is the red line with dots in it, coming back from 1100, and you can see it going up. Doesn't rise that much on the logarithmic scale, but you can see it rising towards 2000. Now, where the lines cross, that's where you can see that surely some of your ancestors were combining. So here we look at it in another way. There's today with you at the bottom, and you go back to about 1200 AD, and you can see your people combining. And of course, you all have to have everybody in that chart for you to be at the bottom. There can't be one of those little dots missing, otherwise you're not there at the bottom. And then after 1200 AD, you can see it shrinking down, of course, because there's not that many people on the planet. And of course, it shrinks and shrinks and shrinks. And the aim, the genetic aim, is to go back to the common ancestor right at the top. So we'll have a look at that. All right, so there's the little group down the bottom, which is the DNA testing for ancestry. When you do your autosomal test, you find out about that little bunch there, which is very interesting. Why is that interesting? Because you find out everything about that bunch that you're seeing there, right up to that top row. And this is called an ascending tree because it starts from you at the bottom and goes up. Now, the partners aren't shown there, they're just shown as one individual there, but each of those is a partner. Okay, sorry, no, wait a minute. No, the partners are shown there, that's all right. Now, let's have a look at the next one, which takes the point of view of one of those individuals on the top row, just one on that very top row. And now we're looking down, and we can see by the same token, assuming each of your ancestors has 2 children, now the partners aren't shown. And you can see them, the children have more children, 2, it's just showing 2. And there you are, look right down in the bottom left-hand corner, that tiny little U, right down there. And that is you. And the autosomal DNA test also finds out, gives you the information of all the other people along the bottom row there next to you. So you find out about all your cousins on that bottom row and the next row up and the next row up. So it is fun for family tree work. And just on this one here, so I've just gone a little bit too far for some reason or another. But anyway, we're looking at this one now. So based on that little group of individuals, you can see that you get a nice little report there. And in my case, Robert Coenraads, you are 64.6 North and West European, 33.5 Scandinavian, and 1.9 Finnish. So that's kind of interesting. That's just the last little bit. Now I just need, can everybody still hear me there? I just a little bit concerned.
Yes,
yes,
yes, we can hear you. Yes.
Fantastic.
Yes.
All right, so isn't this fun? Now each of you can get your own, and that is only $50 or $70 and a little scrape inside the mouth, and away you go. Now, just my little slide mover, I'll just try and make it go to the next slide. Ah, here we go. Now, so that's the cell, that's the DNA inside the nucleus of the cell. Nice and straightforward. And a quick lesson on sex here. You can see the female with a double X and the male with an XY chromosome. Remember, the Y is the short one. And there are the 4 possible combinations of those 4 sex chromosomes in your daughters and sons that will be made up of the different X and Y combinations. So of course, Y is dominant. So the male has the X in there, but the Y dominates over the X, and that's why they're males. And of course, without the Y, then you've just got the double X, and then you're a female. And here again are the chromosomes. You can see the autosomes we talked about, and there are the X and the Y down the bottom that we're talking about. Now, one more little bit of a lesson. Every animal in the animal kingdom has a cell that looks like this. And in the cell, you've got the mitochondria, and that's those blue areas there. They're the power packs of the cell. They provide the energy for running the cell. Now, how did that come about? Here is an evolutionary sequence. You can see right on the bottom left there, an early simple cell called a prokaryote getting its nucleus, and then it's engulfing other things. It starts engulfing other things that it wants to eat, because that's how a cell wall works. It just wraps its flexible membrane about whatever it wants to eat, and it just swallows it in, like in this one here, just sucks it in and eats it into the area there. You can see the nucleus in the middle, and it's sucked in that first purple thing, and then later in it sucks in another one. But it's interesting, when this primitive cell sucked in this anaerobic prokaryote, it realized, giving it sort of thought, which it didn't really have, but it just did it automatically, it realized that this organism that it just ate was giving it more power by being kept as a prisoner in there. And the same with the chloroplast that was swallowed later on in this diagram. So these things became the power packs of the cell, the chloroplast for the plant cell, and the mitochondria for the animal cell. But this is the amazing thing about this mitochondria that's been swallowed and now producing the power. When you analyze the DNA of it, it's got completely different DNA. So it's something else. Now, here, coming back to the sex lesson, there's the father's Y or X chromosome coming in. It's like a coming into a huge planet, but it's the mother's X chromosome is actually a cell. That's a cell, an egg cell with mitochondrial DNA in it. And of course, the sperm is just this little thing, and it doesn't have any, it's not a cell, it doesn't have any mitochondria. So, so here in this diagram, you can see that nuclear DNA from the nucleus, comes in from all your ancestors in that diagram on the left. And the mitochondrial DNA, you can see it's just coming in through the line. It's passed down through the mother the mother's mother and the mother's mother, so that even though the male, in this case, has the mother's mitochondrial DNA, the male won't pass it on. It'll always be the mother. So you get quite a marvelous situation happening here, that by using the sex DNA, you can go back and follow along two specific lines. Using the Y DNA, you go all the way back through the father's line, and you can see the father, the grandfather, the great-grandfather, and they've all got the same Y DNA. And on the mother's side, you're actually going through the DNA in the mitochondria, or the power pack, back through the grandmother, the great-grandmother, etc. So you've got two specific lines, and that becomes, well, in my case, early on in my life, I went back 9 generations. So by going back through my family name, the Coenraads name, through my father and my grandfather, I essentially was following the Y DNA line. And that's the thing that most people, when they do their genetics, they go through that. They go through the father, the paternal grandfather, great-grandfather, etc. The mother's name changes each time, so it's not generally followed, but the mitochondrial DNA follows that line perfectly. Now, I must not take too long here, but now coming back to this diagram that shows us going back through to our common ancestors as the population shrinks, we can again see what an autosomal DNA testing does for us. And we can also do maternal line DNA, that's called mitochondrial DNA testing, and Y DNA testing, and we can follow the blue line and the red line back to the Adam and Eve, to the genetic Adam and Eve, let me say. So it's fun to do. That's another $100 or so for each of those. And they, based on the haplogroups, so let's have a look at Y. We'll leave the mitochondrial just due to time running out here. You can see for me, when you look at all the groups, you can see where the concentration is, and that gives you the source area, dark being the most concentrated, and making these maps of all these different types, with this being one type, you can then reconstruct, and I'll go very quickly through these. In my case, you can see that the Robert Coenraads G has come from the Adam in Africa, and you've gone out through A, B, C, coming up, and there's my line as G. So just ignore that on the following maps, because that's just showing me mine. Now, with what happens is with these, the reason you can follow them back is because of the mutations that happen in them. So in other words, the A gene, haplogroup A, branches off from the chromosomal Adam in this diagram here down at the junction point down. So due to the movement of population or whatever environmental change, there can be a very slight modification to the genes. Now, then you'll have two groups, one that has that's been modified, and one group that hasn't moved, say, that hasn't been modified. So you can see that as they modify, you get haplogroup B, and then haplogroup D and E, and they branch. So once you get onto one of these branching lines, that branch will have everything before it. But when you get off on a side branch, they won't have those things. So you can create the whole web of life starting off from the chromosomal Adam. I wish I had maybe another half hour to explain all this, but better, but we'll just continue on here. So there's this wonderful web, and you sort of know these genetics, these changes happen. And why might they happen? Well, if you go to the north and you need a lighter skin because you're getting out of the hot sun areas and you need to have that vitamin D, there'll be a genetic mutation for you to change accordingly. It's obviously going to be environmentally driven why you change. Now let's have a look at these fun maps. So on this map, you can see starting off with the atom in Africa, and according on this genetics, that's about 235,900 years ago, starting off there. And it's the red line's going out. So again, ignore my G line, that's just going to stay there. So you can see the first migrations there, and the date is over there on the right. And I'm going to go through them now, so you can enjoy seeing what modern genetics has come up with just in this this last decade, really. So that's A, they moved. Then B, 130,700 years ago. I won't read out those times. You can see they've moved in Africa. These are different groups moving in Africa. And then C, shot out. There was an early group of C into Australia, look, 88,000 years ago, and then a surge across into the Americas. Now, we always knew there was something into the Americas 10,000 years ago, but here we're getting such wonderful more detail. We would have never known about this earlier 88,000 years ago. C. Oops. D. Going into the Pacific coast there, into Asia and Japan. E, back into Africa. F, you can't see F, it's the little line running up to G now. And G then was my haplogroup coming out into the sort of the Asia area, one of the first migrations there. H takes a little deviation down across into India, 47,200 years ago. I was one of the first groups into Northern Europe. J, into Europe, but sort of the north of Africa and the south of Europe. K, where's K? Oh, a little branch there. K. Okay, L into India. M into New Guinea. Back curling round into Asia and through Asia into Europe again. O, P, Q. Here we get the 10,000 years ago into the Americas, and R, S, and T. That's the young, the one right into another one into the Pacific. So it's totally wonderful. That's the X. That's Y, and of course X has a very similar picture. So we will we'll conclude there. Thanks very much.
A little time to go, Robert. It's we've got about 4 minutes, so a couple of minutes. Does anyone have any questions at all to put to Robert?
I can see a hand there, I think, from Suzanne. Is that right?
Yeah, I'm curious why they always have like Adam coming from Africa when we know from the Urantia book that it comes from, you know, Middle East, Iran or Iraq or somewhere there, but they always say out of Africa, and I'm curious as to why.
Yes, we'll see what this is. What this is, Suzanne, is that we're actually going way previous, because we talked about 230 odd thousand years ago. So what actually happened is that the humans, the anatomically modern humans, that's where they came from. They came zooming out of Africa there, and then they built themselves up a little bit around the earth. And then after that, you know, the Adams, the Adam and Eve sort of extraterrestrials, if we want to call them that, they were then put into the Middle Eastern area, and it went from there. Now I think if I remember rightly, was that about 38?
Well, I think I was thinking Adon and Fanta. Did they come from Africa then? Adon and Fanta came from Africa.
If I could just intervene there, what's recently happened is because they've discovered the human species. This is to do with geology. Recently, they've discovered some even older remnants of the human race and around India and the Asian continent, and that's only been over the last, I think, 12 months. So they're re-looking at the whole thing, and a lot of this data is based on what we've actually physically discovered. So it's changing over time, and maybe there will be more of a convergence in time with the Urantia book story. Does that make sense to you, Robert, what I just said, or?
Yes, this convergence, because remember, the Urantia book story is only given to us what was known at the time, because we couldn't have any unearned knowledge. We have to go ahead and discover that. So they've given us the best they can, but we do know, that's right, we do know that the humans were there, and it even says it in the Bible story, you know, because Cain and Abel had to do they had to go out and find mates and things like that. There were other humans about, even in the biblical level of knowledge, but clearly in the Urantia book, the implantation of Adam and Eve was a much later thing than the genetic story that I just put on there.
Yep. Okay, so we're getting ready to hand over to the next host and the next presentations. I just want to say thank you, Robert. It's so much food for thought, and we could continue these discussions for a long time, I'm sure. But that was absolutely wonderful.
Well, thank you very much. I really enjoyed presenting that to you, and it's just so wonderful to be together in these COVID-19 times, but we're still all together and talking, and I really love that. And you know, it was a lot to pack into an hour. I'm sorry about that, but thank you very much, and I just enjoyed it. I enjoy all of you. Thank you.